So good morning, everyone. Thank you for joining us today. My name is Helen Chu. I'm at the University of Washington, and this is the next in our series of the Viruses and Vaccines seminar. It is my great pleasure today to introduce Professor Jim Heath, who is the president of the Institute for Systems Biology. Previous to this, he was the Gilloon Professor of Chemistry at Caltech, and in 2018 moved here to Seattle to lead ISB. As part of our collaborative work, he leads the Pacific Northwest Consortium for the NIH Recover study, which aims to identify factors that predict long COVID. And he recently published a highly cited paper in Cell describing factors that are associated with development of long COVID. So it's really my pleasure to welcome Dr. Heath here today to speak to us about his work characterizing factors that anticipate long COVID. So welcome, Jim.
Thank you, Helen. Okay, I'm going to share screen. Okay, is that working?
Looks good.
Great. Okay. All right. Thanks so much for having me talk at this series, Helen. I think it's great that the Bropman Beatty Institute is doing this, and it's terrific to be here this morning. So I'm going to talk today a little bit about that Cell paper, but mostly I'm going to talk about a lot that we've learned since then in terms of expanding upon data. But I'll cover that Cell paper initially. But I want to make sure I highlight all the folks that are involved in this. Of course, there's Helen right there, but a whole bunch of students on my side, a bunch of collaborators from Stanford and elsewhere, and I'll be highlighting some of these contributions as we go through this presentation. Okay, so disclosures. All right, so I think most people by now are pretty familiar with what PASC or long COVID is. It comes across as a myriad of different neurological and respiratory and cardiovascular sequelae that follow a COVID-19 infection. I showed this picture on the right. This is one of the patients that came from the study that Jason Goldman from Swedish and I ran, who was infected with COVID early in the pandemic and ended up with pretty serious neurological issues, and was seeing some demon at night. And this wouldn't have been my choice of how to illustrate the disease, but he tattooed on his arm this demon he was seeing, and he eventually did recover. And this red to green is supposed to represent that journey. But there's a lot of people that don't recover, at least out to 1.5, 2+ years post infection. And I think this is turning into a pretty acute issue post pandemic, which is one of the reasons why Helen and I and several others are involved in this Recover program. Okay, so when we started in 2020, I think in March, we recruited patients that allowed us to look at not only when they were initially diagnosed, but at the stage of acute disease, and then 2 to 3 months later, when they had recovered. And now, of course, we have data that goes farther out. But our goal in analyzing this data, we weren't thinking of long COVID at the time we launched this study. But once long COVID became something that was frequently reported, we analyzed our data at a very deep level, ranging from questionnaires and electronic health records from patients through to very, very deep immunological phenotyping to see if we couldn't associate symptoms with certain risk factors for developing COVID and identify immunological signatures that connected these together. And the idea was that there's probably a handful of risk factors. Symptoms are relatively vague, but these risk factors may actually crystallize what these symptoms mean. And then immunological signatures give you a way to go in and rationalize how these things all fit together. And so the summary of the paper that we published was that we found 4 dominant risk factors that you could see at initial diagnosis of COVID-19, and then a fifth factor that had a different kinetic effect and was associated with gastrointestinal PASC, or PASC is the abbreviation for post-acute sequela from COVID-19. So two of the factors were EBV viremia. So this is DNA signatures of BBV that pop up in the blood at initial diagnosis. And then SARS-CoV-2 anemia, not in the nasal swabs, but in the blood. And these associated with neurological PASC, and we saw them in a reasonable fraction of patients. Type 2 diabetes, largely because we have so many diabetic patients. It's such a common condition, but also cardiovascular issues popped up as well as being a risk factor for PASC, and then autoantibodies. And then finally, we saw that there was the continued expansion of viral antigen-specific CD8 plus T cells that were effector populations, even at the point of convalescence, that seemed to associate with gastrointestinal PASC, perhaps suggesting that there had been virus that had moved into the GI tract. I'm going to elaborate on these two factors that we've been exploring in really quite some depth. So autoantibodies first. So about 2 years ago, we published a Cell paper in which we did pretty deep immunological phenotyping of the kind that I think several groups have done. And one thing that we noted was the presence of these atypical memory B cells, which are an interesting phenotype of B cells that mature, thought to mature in an extrafollicular pathway, meaning they don't migrate into B cell molecules and mature in the normal pathway. And these, at least at the time, had been associated with lupus. Now they're associated with many different things. And so when we looked for autoantibodies, because of these atypical memory B cells, we looked for interferon alpha, which has been reported as dominant autoantibody in long COVID. But then we also looked for these different autoantibodies down here, LASSB, et cetera, that are associated with systemic lupus erythematosus. And what we found using both our cohort of patients and Helen's Harvey cohort of patients was, I think, pretty striking. So what I'm plotting here is a correlation matrix. And so these are the different antibodies against the virus or the autoantibodies that are being correlated with each other. And so you've got viral specific antibodies that are either mature or immature. And then you've got these IgGs against these autoantibodies. And so if you have, antibodies against SARS-CoV-2, of course, is likely you have other antibodies. So you see very strong correlations in this quadrant. And we also saw correlations in this quadrant down here of autoantibodies. I think what was striking and what emerged out of both these cohorts is that, especially for mature antibodies against the virus, we saw elevated levels of autoantibodies that were significant. And the idea, or perhaps a concept, is that the higher the level of autoantibodies, the lower the level of protective antibodies one eventually develops, okay? And so if you looked at these autoantibodies, you found that, in fact, they did associate with, and these are autoantibodies measured at initial diagnosis, they did associate with not necessarily neurological PASC, but with some of these other PASC symptoms that patients were complaining of. Now, this is a pretty tricky area to try to resolve completely. And in fact, and I'll show you that in a minute, that the autoantibodies as biomarkers play an evolving role as one moves through from initial diagnosis out to long-term recovery. And in fact, in our data, in fact, this is like for 2/3 of the patients or so, autoantibodies at the time of diagnosis actually correlate quite strongly with these different certain different past symptoms. If you look at autoantibodies taken a year or two out from COVID infection, and this is work from Akiko Awazaki at Yale, you don't really see a role for autoantibodies at that stage. And in fact, we're collaborating with Akiko to try to figure this out. So we have taken our patient cohort and working with Mike Snyder and Linda Land at Stanford, have actually done a proteome-wide analysis of what autoantibodies associate with specific long COVID symptoms. And this is just an example of that data for fevers and chills, no and yes. And you find that there's, in this case, there's actually two groups of autoantibodies. This data more or less recapitulates the results that we had seen before, but of course with a little less sensitivity on the autoantibodies, but it's proteome-wide. And so it appears that these autoantibodies, well, my typical field that I work in is cancer. And if you have CA-125 elevated and the physician suspects that you may have ovarian cancer, that's a biomarker that says you probably have ovarian cancer. I think for these chronic conditions, it's very, very challenging to find a biomarker that has such ready interpretation because they evolve and have to be interpreted within context. And so I think that's one of the real puzzles in this field is how do we understand how patients are responding by looking for biomarkers in the blood, or how do we understand even what symptoms they have once they've begun developing past, it's almost too late to look in the blood and find those biomarkers. And this is data that we published, an analysis we published in our Cell paper, which kind of illustrates this point. So here's the various past factors we measured: the viremia, RNAemia, this is EBV, SARS-CoV-2, type 2 diabetes, these various autoantibodies. And if you look at initial diagnosis, you find that there's actually quite a few relationships between these, which actually suggests that maybe there is a limited repertoire of drugs that one might use to treat long COVID if you take a leap and say that these may have at least the flavor of causal-like factors. Once you go a week later, these relationships are substantially diminished. And once you go out to recovery, these relationships are actually gone. And that just says that all patients are starting at a slightly different point. There are different ages, different sexes, different weights, different health conditions, what have you. Over time, they all just evolve on different trajectories. And in fact, you can model this, and it's kind of the expected answer using chaos theory from physics, but it's actually at the heart of why this understanding these chronic diseases are so challenging, especially without a kinetic window on these patients. Okay, so I'm going to talk about now what we've learned about antigen-specific T-cell responses, and back up and give a little bit of background from the Cell paper, and then talk about what we've done since then. So there were early reports that Epstein-Barr virus antibodies seemed to have an association with long COVID symptoms. But when you are looking in the blood at antibodies, they will last for a long time, and it could be, even out to years. And so it's actually very difficult to know if these antibodies against Epstein-Barr virus, if there was a triggering event or what have you that was associated with the infection. And so a more quantitative correlation of these virus antibodies and their origin with COVID is by looking directly at the genetic material of these viruses in the blood, because that basically is going to be very, very transient. And so we worked with Josh Hill and then here at ISB, Kai Wang, to look for not just SARS-CoV-2 RNA in the blood, but also evidence for reactivation of Epstein-Barr virus and cytomegalovirus. And here's the data that we saw. So in about 20% of our patients, give or take, at diagnosis, one actually sees a pretty strong signature for both Epstein-Barr virus and for RNA-emia of the SARS-CoV-2 virus in the blood. That goes down quite a bit within just 1 week, and at the time that you 2-3 months out, it's just barely detected. And now the SARS-CoV-2 RNA-emia is probably also a surrogate for just disease severity. And you can see that here because, and this was reported in a Nature Com paper last year, that SARS-CoV-2 viral levels in the blood are actually predictive of mortality. But this EBV says that infection with SARS-CoV-2 is actually triggering a reactivation of Epstein-Barr virus, which typically harbors in B cells. Okay, and in fact, you see that these viral levels associated with neurological PASC pretty clearly. Okay, so T cell responses against these viral signatures. What one normally expects to see in a T cell response against an active virus is that you will have a naive pool of T cells that will, some of those clonotypes will expand to form effectors. And then as one recovers from the disease, those will contract back into, likely memory phenotypes. And so we worked with the folks from Adaptive Biotech to do what they call as their mirror assay, which is you just expose PBMCs from these patients to peptides that are derived from the virus, and you and look for evidence of antigen stimulation, and then you identify the T cell receptor beta chain. And so if we take all of the T cells from all of our patients, like 1 million T cells here, you get this UMAP, which many people are familiar with. You have naive all the way to effective populations out here. And then we project onto this UMAP the T cell that are specific to the virus, to SARS-CoV-2. You find that, in fact, by and large, it behaves like what you expect it to behave. We also found evidence for CMV-specific T cells. Now, there is no CMV reactivated. And so what you're looking at is what's called bystander activation of CMV. So it turns out for most of the patients, you see an elevation of these T cells into effector populations that then decays out to convalescence. But for a subset of patients, those effector populations continue to increase. And that subset very clearly is reporting gastrointestinal PASC, arguing that GI PASC is a set of symptoms that has just a different kinetics associated than other PASC. Okay, so that's a pretty incomplete story that we wanted to dig into more deeply. And so what we wanted to do was to look for the entire viral proteome-wide analysis for both SARS-CoV-2 and we looked at CMV as well, in which for each T cell, we have the paired alpha beta gene so that we actually know which T cells are clonally expanding and which ones are not. We have the peptide antigen and the MHC allele, and we have the single cell RNA-seq phenotype. And that we do this for lots of patients in parallel. So this is a pretty challenging experiment. And we had to develop some technology to do this. And we worked to validate these technologies with John Lee and Roland Strong and Phil Greenberg. And I'm going to give a shout out to my students and postdocs, Jingyi, Zhongchen, William, and Daniel, who did this work and are doing it. So the challenge, if you want to capture lots and lots of T cells, the standard capture agent for T cells is a peptide MHC tetramer that Mark Davis developed 40 years ago or something. Those are unstable. They're typically folded in solution, and you get you can do a handful at a time, maybe 2 or 3. You can't do a whole viral proteome work. And so we explored this alternative technology in which you build the antigen. This could be a neoantigen in this case for cancer, but this could be a viral antigen. The MHC HLA subunit and the beta-2M subunit, all as a single molecule. This is called a single chain trimer, and it's been reported for a few years. But we extended it into a library format and then explored a whole bunch of mutations to stabilize this structure of this antigen-presenting MHC unit. And so this whole unit here, we engineer it to be secreted from cells and secreted from mammalian cells. So it typically contains the same post-translational modifications that a naturally occurring peptide MHC would. But of course, it has these linkers. And so you don't know how realistic this thing is compared to a normal peptide MHC. So anyway, we make this big library, we test it, and this is just looking at I'll show you a lot more data. This is like CMV specific clones captured using this technology and a refolded peptide MHC. This is the gold standard technology. And you actually capture more or less the same clones. In fact, you capture exactly the same clones, except for this one is captured by the SET, and this one actually exists in public databases for CMV. So basically, these two are relatively equivalent. Okay, so this now gives us an approach for building a whole library across the entire SARS-CoV-2 proteome of T-cell antigen-specific T-cell capture agents. And that's what we did. This just shows about 1,000 of these elements in plasmid form. And when you express them, this is the coverage you get across the SARS-CoV-2 proteome. So here's the different, for example, non-structural units. Here's the spike. Here's some of the envelope proteins, et cetera. And so we have pretty good representation for HLA A2.1. That's what this is here across the whole viral proteome. We also did B701, B702 SARS-CoV-2 antigens, B702 CMV antigens, A201 CMV, and EBV antigens. So the whole library, everything put together, was about 650 of these. Okay, so the way that we analyzed this, used this library, is that we took 65 COVID-19 patients at the point of acute disease who were HLA 2.1 from our own database. And then we split this 650 element library into 10 batches, and we would make using a dextramer with DNA barcodes. We would capture these T cells and then flow sort them and then hold them on ice. And so we had this whole workflow designed so that it only takes a couple of hours, and you peak the cells on ice, and then you move them through a 10x type analysis. Okay, so in the end, what you get is an entire UMAP of these antigen-specific CD8 plus T cells, in which for each individual cell, we can identify, based upon the barcoded dextramers, what the antigen specificity of it is. We had the T cell receptor alpha and beta chain. And then for each of these patients, we had the whole genome. And so using SNP analysis, we can actually assign each one of these individual cells to one of these 65 patients using the whole genome. So it's a lot of computation, but in the end, you have a completely new way to look at antigen-specific T cells. And when you look at this UMAP, you have the same type of UMAPs one expects to see, going from naive back here, here's proliferative up here, all the way to exhausted down here. Okay. So the first thing we see is we can project for a given antigen. Many patients tend to respond to many of these antigens. And so this is antigen-specific responses against this particular SARS-CoV-2 antigen, and the different colors are different patients. Here's a CMV antigen, and here, once again, different colors are different patients. And one can see from just these two antigens that they may occupy, slightly different parts of the UMAP. But when you put all the data together, you find that the SARS-CoV-2 T cells are really quite distinct on the UMAP than are the CMV specific T cells. So these bystander activated populations are really very different in terms of their phenotypes. If one looks at different patient responses against the viral antigens, one finds that in general, stronger responses, if I just ordered them from stronger to weaker, that stronger responses to weaker responses anti-correlate with disease severity. And you can see that this is disease severity at 1 week and at diagnosis, and that this slope is positive for both of these as this goes down. But basically, there's a lot of noise here. I mean, if you look at, it's not experimental noise, it just says that if you have patients that are moderately infected, they're kind of all over the map, okay? And then the phenotypes of these T cells also changes as one goes from strong responses to weak responses. So this is the map of basically the populations we saw and the populations that clonally expanded. So this is the number of T cells per domain. Here's the individual domains cut apart into the antigens that captured T cells. And then the thickness of these bars is the clonally expanded populations that we found against specific antigens against these domains, okay? So this one up here is a massive response. Every single patient pretty much sees this antigen against ORF6. And it's equivalent to, for those of you who know CMV, there's a PP65 antigen that is pretty much seen by everybody. This is the SARS-CoV-2 antigen that is like that, okay? And there are many populations of T cells that are strongly clonally expanded against this antigen. And then other strong responses as well with clonal, but this is the strongest. In fact, I think I've even thinned this a little bit to get everything on the same page. If you clone T cells that you capture this way, and you test them against antigen presenting cells presenting the correct antigen, they will kill the cell. And so what we're finding, you can close the loop and say that these T cells that we're finding this way, these clones, actually do detect the antigen, they are activated against the antigen, and they participate in antigen killing. And so this is just a killing assay, but this is just for a whole bunch of spike-specific T cells against 3 different HLA alleles, showing that they have some level of activation when they see their cognate antigen. And when you expose them to a dummy antigen or irrelevant antigen, they do not respond, okay? So this story gets very interesting when you start looking at cross-reactive T cell populations. So what I've plotted here are all of the antigen specificities for which we detected T cells versus the same thing. And we're looking at T cells that react with, we're looking at this a cross-reactivity map. And so most of the T cells are only reactive against one antigen. And that's what these diagonals are. But there's a set of T cells down here in the bottom left corner that are cross-reactive. And if we blow this area up and we look at cross-reactivity versus clonotype expansion, one finds that, in fact, the T cells that are most clonally expanded are also the ones that are most cross-reactive. Okay, so that's, I think, a pretty surprising finding, but one that comes across with a very high degree of fidelity. If we look at this, many of these clonally expanded populations are against this antigen here, ORF6. If we look at these clonally expanded populations against ORF6, and you plot them against just small numbers at CORF6 to large numbers at CORF6, it almost appears, in fact, it kind of appears, that cross-reactivity sort of is the switch that turns on. In fact, many of these populations see ORF6 with no cross-reactivity, but if you have enough expansion, you have cross-reactive populations, okay? And the cross-reactivity is not all to all. In fact, the cross-reactivity is very specific. So if the blue here, for example, is the same antigen that is cross-reacting to in all of these things. The purple is the same antigen, et cetera, okay? The cross-reactivity T cells occupy a different set of phenotypes than do the T cells that have low cross-reactivity. That's suggesting that as these T cells expand, as they clonally expand, their phenotype is evolving. Now, there's a paper that was published that I'm going to connect back to in a minute. We were involved in this work, but it came out of Mark Davis's group at Stanford that talked about this novel type of T cell that expresses what's normally thought of as an NK cell-like receptor, KIR plus CD8 plus T cells, that he identified as suppressing pathogenic T cells and active in autoimmune diseases and COVID-19, okay? If we take our data set and we look at those T cells that are most cross-reactive, so we make a cross-reactivity metric, and we look at what genes are turned on as they become cross-reactive, we find these KIR genes get turned on with very high fidelity, also looking as if they're getting turned on as a switch, meaning nothing gets turned on until you get a certain level of cross-reactivity, and then it's on, okay? There are a host of other NK cell genes that also get turned on, okay? And in fact, these cross-reactive T cells have a lot of other transcripts that are turned on. So these are genes that are most commonly associated with NK cells, but also you occasionally do see them in T cells, but they give these cross-reactive T cells definitely an NK cell-like flavor. And so here's just some of the genes that separate the cross-reactive T cells from the non-cross-reactive T cells. And if you analyze these genes, you find that you get an increased NK cell-like character. You get an activated signaling pathway that seems to pass through cytokines or a toll-like receptor, and you get an activated apoptotic pathway. So I believe what's happening, and we're cloning now a bunch of these to show whether I'm correct or not, is that these T cells are cross-reactive not because the T cell receptor is cross-reactive, but because they are expressing a new receptor on their surface. And in fact, some of these are binding MHC class 1 that is giving it this cross-reactivity. But also, you've got an activated apoptotic pathway. Okay, so we see all these things happening. It's not clear what follows what, but I think there's a couple of hypotheses that one can put forth that might explain this data, okay? Number one, I think that a successful virus will tend to distract cells within your immune compartment so that you go after irrelevant targets. That's what this CMV activation is about. And from what we can tell, there is not a great benefit in the ORF6 T cell response either. There does seem to be benefit in NSP response. A successful response against a virus typically has a broad diversity of T cells. And when you have T cells that are very strongly clonally expanding, there seems to be a safety mechanism that shuts them down. That safety mechanism has as a byproduct this cross-reactivity, but it also has this apoptotic signal. And if you look in our data set, you will find that the same T cells that are attacking the virus at acute disease are not the clones that remember the virus. And so some of these highly effective populations that clonally expand simply do not survive. And this transition, this cross-reactivity to an NK cell-like phenotype with some apoptotic signaling is probably a defense mechanism to shut down that response, okay? The other things I just want to put forth is that understanding the role of the past factors play through the disease journey is critical, I think, to understanding past. It's very, very challenging to look late in the disease course, late in, when patients are already reporting these past symptoms, and back up what were the causative agents there. A comprehensive analysis of these viral antigen-specific T cell responses across an entire viral proteome and across many patients gives us a fundamentally new set of eyes on trying to understand antiviral immune responses. And I've tried to highlight that at the end. This is a brand new data set, and we've only had a couple months to go through it, but it's already really changing the way that we think about T cell responses, and even changing the way that we think about selecting antigen-specific populations for cancer immunotherapy applications. And with that, I'm always looking for talented postdocs. So if you are a talented postdoc, please contact me. I want to thank you for your attention, and I want to acknowledge all these various funding agencies, and I'm happy to answer any questions.
Great. Thank you so much, Jim. I think people can go ahead and raise their hand, or if you would prefer to go ahead and put the questions in the chat box. Okay.
I don't see any questions at all.
I don't either. Maybe I'll go ahead and get started and ask you one, and just ask you what your thoughts are on sort of the overall trajectory of long COVID over the course of the next several years, and where do you anticipate things going?
Well, I think as you are finding, it's not, for a lot of patients, it's not going away. Right now, the big recover study that you and I are involved in is, it's really about analyzing and defining long COVID. There's no opportunities in that study to treat patients. And I think there has to be, I suspect there's going to be a number of clinical trials that would be tried. I think some of the ones that have been put forth, and some of them suggested by our paper, like antivirals and things like this, will tell us something. But we know there's clotting going on in many of these patients that happens in long COVID. We see in our data that we can associate early time autoantibodies with later time clotting factors. That's a very, very strong association. And we also suspect this clotting may be occurring through non-standard pathways, through the lectin pathway, coming from ongoing endothelial cell damage. That suggests certain treatments. There's some metabolic treatments that have been initially tried with moderate success by this company Excella, I think. But I think all of these trials will have to be very deeply studied to understand what the therapy is doing. We may find that some of the therapies may have an effect, but that effect may be subclinical, but it doesn't make it useless. It may be something that would be more effective in combination. But this is a big looming issue, and I think we as a country are trying to get on top of it, but it's a challenge to get on top of it.
Yeah, I mean, I think that what's particularly concerning is the lack of randomized clinical trials on a large scale for potential therapeutics. So hopefully that will come. There are now a lot of questions for you. I'll start here at the top with a question from David Gordon. Have there been specific certain COVID strains that have resulted in more long-term COVID than other strains?
I think it's right now probably more anecdotal than anything else, but it looks like very likely the Delta variants and pre-Delta are probably a little worse. Now that's coupled with the fact that many people got vaccinated at around that time. There is some evidence that Omicron strains do lead to long COVID, but because of the different mechanisms of infection of Omicron and pre-Omicron strains, it's for sure going to be different in terms of the types of symptoms that evolve. And it does appear that, you know, even for patients who had been vaccinated and got Delta, you could get long COVID, but the risk was reduced by like a factor of 2. And my guess is for Omicron, it's reduced even further, but it still may be there still may be a constant source of patients with long COVID from these Omicron-like strains.
Yeah, it does look like the numbers are going down, or the proportions are going down over time. So that's reassuring, at least. Lauren Rhoda has a question. Did you see or measure any markers of lung damage or fibrosis? And is there still a role for damage in causing long COVID symptoms, or is that less likely now?
We saw markers of endothelial cell damage that continue. So I can't remember what the protein is, but there's a protein that's associated with endothelial cell damage that you can find in the blood. And in patients that are reporting at least a host of different symptoms for long COVID, that marker does not seem to be returning to normal in a number of these patients. Doesn't mean it doesn't eventually get there, but we're looking at that as a way to assess whether these therapies against the non-traditional, the lectin pathway of coagulation would be relevant. And you do find evidence that it would be a relevant treatment to try from that marker.
Great. Question from Noah: how do vaccines play into the development of autoantibodies and PASC?
Well, they lower PASC. Whether they have a role in autoantibodies, I think, is unclear. But they certainly, you know, give you quite a bit of protection against PASC. So I would be surprised if they actually have a major role in the autoantibodies. I should also put out a comment here. So our autoantibodies were measured by an ELISA type assay, which is really quite a bit more sensitive than, for example, if you had a LabCorp measurement of autoantibodies, which is a pretty common thing to do. If you have LabCorp do a quantitative panel of autoantibodies, it's probably not that different. But many of us on this call will actually have autoantibodies in our blood. They're at subclinical levels, meaning they don't give you lupus-like symptoms or other symptoms that may be associated with high autoantibodies, but they actually can play a role in PASC. And so some of the discussions that we're having with the Yale group and others is for some of these antibodies, you know, what is the right level? What is the right way to measure these things and report them? Anyway, it's an ongoing discussion, and I don't think it's 100% clear yet. I think you measure them as sensitively as you can, and if it's informative, great, and if not, then at least you know.
Great, thank you. A couple more questions. Megan has a question about whether people living with HIV were included in your cohort studies.
They weren't excluded. I think we might have just had 1 or 2 patients that had HIV, that were HIV. Now I don't know in your study, Helen, if you had such patients. I know in the National Recover study, there must be quite a few.
Yes, we again also did not exclude, but we also, but none per self-report.
Right.
Yeah.
Because of the immunosuppressors that some of those patients tend to be on, it could be and we would have to go back and really dig, but my guess is that there's an impact.
Yeah. Yeah. We certainly had a lot of people who were immunocompromised. So
Yeah.
a question from Nick. If the T cells you referenced are not going through the follicular pathway, where are they expanding?
I think they're expanding the B cells. The B cells is the atypical memory B cells, which is what I referred to as being extrafollicular pathway. I guess they happen in either non-B cell follicles in the lung or the blood. I'm not a B cell expert, so I just know what I read. We have actually we've done quite a bit of studies of these atypical memory B cells, and in fact developed an entire phenotypic map of different atypical memory B cells and how they evolve. Basically, it's a pseudo-time type trajectory for those of you familiar with what one can do with scRNA-seq. And it's complicated. I'll just leave it at that. It has an evolving relationship with PASC over time, which is probably not that surprising given that all of these almost every marker, every single one of these markers has an evolving relationship over time. It's what makes this so challenging.
Okay, a couple more. I think these two combined together. A question from Denise and from Corey about therapeutics. So first up, the question from Denise, which is will there be a replacement for Evusheld anytime soon, with it being not as effective against newer COVID variants? And the second question is around antivirals and valacyclovir, and whether or not that might help with PASC, or is it too late with the immune response?
Yeah, the first question, I don't know. The second question, and there's a very big national debate right now ongoing on what types of drugs, which drugs will be done in what trials, the size of studies. I think actually a lot of 200-person type double-blind studies are really called for here to try to work through this. We'll find out. Antivirals, I think that they can be very effective, but I think you have to use them really early. And that was the message that was, illustrated by in our results very clearly. And so when patients are reporting long COVID symptoms, they may be 6 months out from infection, or 3 to 1 year or so. My guess is that antivirals aren't going to be very effective. They may have some role, but I don't really see it. And on top of that, you've got EBV and SARS-CoV-2. If they're somehow tissue resident, those are 2 different viruses. One's RNA, one's DNA, different antivirals. So there you go. It's just not an easy answer to that unless you can use these very early on.
Okay. Another question, which is somewhat related, and I think you've partially answered this, is a question from Josh Schiffer. Based on the complex kinetics of the antibodies and other potential mediators of PASC, do you think there's an optimal time to intervene? Is there a role for treating high-risk people in the early window after acute infection?
Yeah, my guess is that the best time to intervene, as with all diseases, is as early as possible. And there are some indications, for example, that anti-clotting medications that hit the lectin pathway, and this is not exactly double-blind rigorous trial, but there were some indications that may actually be effective in preventing PASC if it's used early. I think these antivirals early. I think the real challenge in developing effective drugs is going to be this is why these studies need to be deeply characterized, is that it's really hard to have, metrics of that associate with these symptoms. And so looking at the effectiveness of these drugs, we desperately need biological metrics that associate with symptom reduction or not. And I think that's a big challenge with all these therapies.
Great. It looks like we have probably time for just one final question, which is from Noah. And it is, what is unique about SARS-CoV-2 leading to PASC as opposed to other coronaviruses from the same family, or I guess other viruses in general?
Well, I think the major thing that's unique about it is that it was, from a viral perspective, it was such a successful virus that PASC affects just a much larger population of people. And we have had those individuals, those patients and participants donate the time and blood into studies like what we're doing that help us begin to understand this. I think some of the issues that we're finding here with SARS-CoV-2, such as EBV reactivation or bystander activation of CMV T cells or autoantibodies or whatever, are probably the kind of things you would find in a study on a bacterial infection like Lyme. You'd probably find it perhaps even in post-acute sepsis type conditions. But it's really hard to get 300 or even 17,000 patients like in the RECOVER study that would allow you to really nail these things down. And with SARS-CoV-2, we've actually had the patients, and we're infinitely grateful to them.
Yeah. I mean, I think the challenges, of course, are precision, right? So yeah, with a lot of these studies.
Right.
Okay, well, thank you so much, Jim. It was great to have you today. It was so interesting to hear about the work, and just one quick plug for next time. So next time we have Cecile Vaboud who will be speaking. She is a scientist at NIH at the Fogarty Center, and her work is on disease modeling for respiratory viruses. And she will be presenting some work related to the Seattle flu study, as well as other work, I believe, from South Africa. So we're very excited to see her next month. So thank you so much, Jim, and thanks for everyone for coming.